Hello and welcome to this week's episode of Research Goes Viral. I am the host for today. My name is Stephen Devlin. I'm a PhD student here at the Centre for Virus Research, and I am surrounded by a plethora of guests who are going to introduce themselves in just a moment. But today we're discussing a really exciting paper. And I know you're in the room with me, but genuinely did enjoy reading this paper. I thought it was beautiful, really nicely laid out, told a good story. And I'm very excited to do a deep dive with you today. But before we get into that, if we just go around the room and just introduce ourselves, our position, and kind of what we did in relation to the paper. And if we start with yourself.
Yeah. Hi, everybody. Thanks for listening. I'm Antonia Ho. I am an infectious disease clinician and a clinical senior lecturer at the CVR. And I led the research investigation for this paper.
My name is Vanessa, and I work as an experimental pathologist. So my topic is looking into the morphology, how do the cells look like, and we also work on where the virus in the cells actually is located to give it in a context, basically, in the disease. Yeah.
And my name is Richard Orton. I'm a bioinformatician, and I was involved in analysing the sequencing data, the metagenomic sequencing data, to identify what viruses were present in cases versus controls.
Perfect. So yeah, welcome everyone. Thank you for agreeing to sit down and talk with me today. So as I say, I thought it was a great paper. I thought it was a really compelling story that was laid out, and I think the data is fantastic as well. And just reading through it, I was great paper. So what is the paper we're talking about? Well, it is called, or it's titled rather, adeno-associated virus 2 infection in children with non-A to E hepatitis. And so to get kind of background in what led up to this paper, if we go back to April 2022, and basically a collection of children presented at clinic with hepatitis. And that, was the start of this whole investigation. So do you want to talk us through kind of how that all started?
Yeah, I mean, this actually started on the 31st of March 2022 when a very astute pediatric liver specialist called Rachel Taylor, who's one of the first authors of the paper, she noticed a kind of cluster of children who presented with jaundice, so yellowing of the skin. And when they were admitted to hospital, they find that they had very high levels of liver inflammation. So that's what we call hepatitis. And normally in an average year, you would notice maybe 2 or 3 of these cases where you have hepatitis, but the underlying cause isn't found. So, you usually screen people for the kind of most common causes, usually either a virus or drug, toxin, or autoimmune, which is when your body thinks that your organ is foreign and attacks it. So you have a kind of abnormal immune response. But none of these children had any of these common causes. So she had noticed at least 5 cases, so alerted Public Health Scotland, and they actually contacted the other liver units and also found similar cases in young children. And therefore they thought, there's something abnormal here. So, an outbreak investigation was instituted. Public Health Scotland was involved, and UKHSA as well, because it turned out that it didn't just involve Scotland. There were cases across all 4 nations. And I lead the recruitment for the ISARIC, the WHO ISARIC CCP-UK, the Clinical Characterisation Protocol study, where we activate for any emerging outbreak to allow us to recruit patients and take research samples and do tests that are not routinely available, for example, like the sequencing. So this is how this investigation started.
Perfect. And so ISARIC, that's the International Severe Acute Respiratory and Emerging Infections Consortium, if I've read correctly. And so who forms that body, and how does that sort of operate on a ground level?
So ISARIC was set up really after the 2009 influenza H1N1 pandemic, because they noticed that, when there is an outbreak, everything is quite chaotic. Everyone's kind of scrambling to understand a new disease. And there's no kind of, in order for people to have standardized comparison, you kind of need a protocol, that is approved in advance so you can activate quickly, because any one of us who've done studies that involves recruiting patients, ethics approval take months to do, by which time it takes such a long time that you often miss half the cases. But also having a standardized protocol that can be used for any emerging infection and adapted to that disease, then it means that you can compare it between different study sites, different geographical locations as well. And so this was set up, and it's been activated for a number of emerging outbreaks, including monkeypox, Zika virus, MERS coronavirus, and most recently for COVID-19 as well. So in the UK, we recruited, I think, over 300,000 patients for COVID. And just gives you a power to really understand new diseases, how they impact on the body, immune response, virology, and it allows just obtaining data itself, but also allows you to obtain kind of biological samples. So, Dr. Taylor alerted Public Health Scotland on the 31st of March. We activated on the 2nd of April and recruited our first patients within a week. And in fact, the first samples from these cases were sequenced, I think, by the 7th of April. So within 1 week, we had kind of first tests that were done on these cases. So it really kind of highlights how important studies like this are, that, speed is of the essence, particularly when we're talking about children who are really sick with hepatitis. And a number of them, not only were they admitted to hospital, but a lot of them were very sick. And 15 children in the UK required a liver transplant, which is very serious. Yeah,
absolutely. And I think that's an important point to hit on there, is the speed of that response, because even getting, normal lab work done can take a wee bit longer than a week, but that's incredibly quick. And that's, as you say, very important when you've got these new emerging things coming out. You need to get as much information generated as quickly as possible so you can formulate some sort of a plan to what are the next steps. And so that kind of leads on to the next question of, so you got these samples almost within 2 days, and then kind of what's the next step in the ISARIC protocol of generating, data and looking at these samples?
Yeah, so I mean, apart from analyzing samples, you have to try and understand, first of all, for a kind of outbreak investigation, you need a case definition. So the kind of understanding the epidemiology. So we knew that the majority of children were under the age of 10. So the kind of UK 4 nations criteria for we included children under 10. The first cases, I think, dated back to March. So we kind of put a timeline that, most of it was children presenting with this syndrome from the beginning of January. We noticed that the degree of inflammation were quite high. So normally your liver enzymes, we test AST and ALT, should be less than 50. And these children, they were up to 2,000. So it's really marked. And I'm not a pediatrician, but I've been told by Dr. Taylor and other pediatricians that degree of liver inflammation is very, very abnormal. You then do a very detailed workup. So the routine clinical tests, not even just the research investigations. You screen for all the causes that I mentioned earlier for for viruses, like viral hepatitis A, B, C, E, Epstein-Barr virus, herpes viruses. Screen for kind of drug exposure, but you also do a very detailed questionnaire, particularly we call it a kind of a trawling questionnaire, to see if there are any other particular risk factors or exposures. For example, foodstuffs, could be new medication or travel, any geographical and you look for whether any kind of siblings are involved, people from a particular geographic location. And really nothing was turning up for these children, just that they were mostly young children. Majority of them didn't have any underlying health condition. So you kind of cast a very wide net, test for lots of things and don't make any assumptions. but it was felt that an infectious pathology was most likely, which is how we kind of progressed very quickly to sequencing, because, that allows us not to make any assumptions about what is the underlying bug. And I think Richard can go into a bit more detail about metagenomic sequencing. But and this was quickly agreed because we had all these research samples that sequencing was top of that list of a battery of tests that we did.
Okay, perfect. And that's going to lead us quite nicely onto our next section where we talk about metagenomic sequencing. Now, I'll put my hands up. I am not an expert in metagenomic sequencing, but we have a man here who is. So kind of what was the... tell us a wee bit more about the process that you undertook when you received those samples and how that worked.
Okay, so I'm on the computational side of it. So there's a whole enormous bit of lab work that has to go on first, so I can give you my sort of understanding of it. Apologies to everyone who knows. So once you get the sample, and I think the sample that they normally receive is nucleic acid, so either RNA or DNA. It's received into the sequencing lab, or probably into the biobank, and then go to the sequencing lab. And then essentially what metagenomics is you're trying to sequence everything that's in the sample, really. But there is a bit of a scale issue. You can't sequence every single piece of DNA in there because there's just far too much of it. So you are essentially just taking a sample of that. So as a result of that, sometimes metagenomics may miss something that's a really low level. So it's not a completely foolproof way of doing things. If something's there at high frequency or reasonable frequency, then metagenomics should identify it. What you're doing is you're just sequencing all of the either DNA or RNA that's in the sample, and then once you've got all that sequence data, you can compare it to databases of things. So the guys in the genomics lab, they'll get the sample and then has to go through what's called library preparation, where it's sort of you're fragmenting the DNA and RNA to the correct size, goes through lots of checks to check it's good quality. There's kind of adapters ligated onto the ends, and that gets put into the sequencing machine, which essentially sequences all these pieces of DNA and RNA, which is by that point it's DNA, and then which creates all this sequence data and gets handed to us in bioinformatics. So what we get as a kind of raw input is just lots and lots of typically short DNA sequences, millions and millions for each sample. So the objective then is to kind of say, well, what is all this DNA? Is it just all the human host? Is there a virus in there? Is it bacteria or something like that? So at the very start, I think we had five patients, 5 cases, I think, at the very first run. So we had 5 samples, and they went through an RNA library preparation and then also DNA library preparation just to cover the bases. And maybe there was something that was an RNA virus, say DNA virus, just to cover it all. So we sequenced that, and then we kind of analysed the results. So this was what Tony was saying about. This was quite quick. I think this was like within a week. So this was just our first attempt to see what was in there, and then kind of pretty much straight away within a few hours you could see that the only thing that the samples had in common was this virus called AAV2, which we'll go on to later on. But there wasn't really anything else in there in terms of viruses. Certainly in all of the samples, there was nothing shared apart from that AAV2. There was hints of other viruses in there. One had a hint of an adenovirus, which before we started was hinted at this could be the cause of it, which is adenovirus 41 is what people have thought might be involved. There was sort of a hint of that in one sample. There was herpes virus hints here and there. But certainly at that first run, AAV2 jumped out. And I certainly had never done anything with AAV2 before, so everyone was immediately Googling it, what on earth is AAV2? But then that initial 5 sample run led to then more samples and more checking, and then trying to get controls in to see if, because it could have been that AAV2 was just everyone in the population, and therefore wasn't important.
And I think, correct me if I'm wrong here, AAV2 typically is asymptomatic, so that must have been it doesn't usually cause pathology, as far as I'm aware.
Yeah, I
Typically.
mean, I think most people didn't know, including myself, knew virtually next to nothing about AAV2, which stands for adeno-associated virus 2.
Oh yeah.
And it's from the genus kind of Dependo parvovirus. Why it's called Dependo, it's because it cannot replicate by itself. It needs a helper virus in order to replicate in itself. And helper virus is typically, given that it's called adeno-associated virus, adenovirus is one such helper virus. There are different other types of helper viruses, including herpes viruses, human papillomavirus, Bocavirus as well. But it does require co-infection with another virus in order for it to multiply. It's not known to cause human disease. It's probably best known for being a vector for gene therapy, which is used a lot in children. And although it's not really widely described in literature, actually, people who work or give gene therapy recognize that they do see hepatitis, and quite frequently they have to treat prophylactically with steroids, and there have been rare cases of severe hepatitis associated with AAV vectored gene therapy.
Okay. So was that kind of a light bulb moment when you saw that flag up, or was that just let's pin that to one side and
I think it was a bit of a light bulb. It was a light bulb, but it was a very cautious light bulb.
Yeah, there was a bit of head scratching, and you always have to be, a bit cautious about what you find. Like, can it be explained biologically? Has someone ever described that before? Could it be one of the controls? Because a lot of the initial clinical samples we got were from samples at the West of Scotland Specialist Virology Centre, and they'd done the extraction for us, and we wondered whether it was actually a control. But we actually found it in all 9. So we ultimately spent a lot of time doing the detailed metagenomics in 9 patients, and we found it in plasma of all 9 cases, and also liver samples from 4 out of the 9 patients. So that gave a pretty strong signal. As Richard said, we also found adenovirus, I think, in 6 out of the 9 cases. And in the kind of routine lab testing, adenovirus was probably one of the most common viruses detected by routine samples. So that's usually PCR in respiratory samples or blood samples. But not all children had it. And it might just be that because we were picking up patients in March to April, July time, it may be just that we were picking up what was circulating at the time and had nothing to do with it. So in order to say whether this AAV2 is important, you need to recruit controls. So controls, we ended up recruiting 4 different types of controls. So ideally, you want them to be matched by age, by time, by geographical area. But, we're talking about blood and liver samples, which healthy children don't usually have that taken routinely. Adults more commonly, particularly blood samples. So we recruited 4 different groups of control. Groups 1 to 3 were from a study called Diamonds, which had been recruiting since the beginning of 2020 to look at causes and molecular signatures of children with fever. So we were lucky. So group 1 are healthy children to see whether you would routinely find AAV2 in healthy kids. Group 2 were children who had adenovirus infection but didn't have hepatitis. So, would you find AAV2 in kids that have adenovirus? It's just something that we never tested for AAV2 before. The third group were children that had hepatitis for other causes, not adenovirus, to see, is it just something that happens if you have hepatitis and you reactivate AAV2? And the fourth group, which is really important because the first 3 groups of children were not necessarily recruited at the same time as our cases. And we wanted to see, it was AAV2 just circulating in children. And so we recruited what we call contemporaneous controls at the same time in the same age group and in Scotland. So we had not many, but we had 16 samples from children that were attending hospital in March and April at the same time as cases to see if we can find AAV2 in those. And essentially, we didn't find AAV2 in any of the group 1 and 2 controls or any group 3 controls. We did find, I think, 4 out of 16 of the contemporaneous Scottish controls, suggesting that AAV2 was circulating in Scotland around that time, but that high levels of AAV2 is not common. It's certainly not in healthy children or children who have adenovirus infection that without hepatitis or hepatitis for other reasons.
Okay. Because one thing I thought about the paper in general is that it was really well controlled. And I can understand the rationale behind each of the control groups very easily to read it. As someone who's not a pediatrician or someone who studies these things. So when you talk about the case control, that is essentially just comparing your cases where you say, right, this is the epidemiological, pathological occurrence that we're looking to study, and then you're controlling that with other members of the population who have certain features of that, but not all of the features, so you can narrow it down. So again, that's the next point of evidence. There is that when you start thinking, okay, AAV2, this is starting to look like this might be the causative agent, or when does that really start to become I think, this might be the...
I think you just gradually build layers of evidence, right? So when we talked about the metagenomic sequencing, that's only in 9 cases, which doesn't give you much power to say anything. So by this point, we had recruited over 30 cases. So we basically expanded our investigation. Metagenomic sequencing is laborious and needs very lot of specialist techniques, but it's also very expensive. So once you have a pathogen of interest, you can then develop a molecular cheaper test like a PCR. So what we did was we switched to PCR and did AAV2 PCR in 32 cases. So the paper mostly includes 32 cases and 74 controls, which is from the 4 groups. And we found AAV2 in 26 out of 32. So that is 81% of cases. And remember that the definition for a case is quite broad. It's just a children under the age of 10 presenting from the 1st of January 2022 with deranged liver function above 500 of AST or ALT. So you will inevitably pick up some that are not cases of this kind of phenotype. But in the controls, we only found it in 5 out of 74, so only 7% of controls. So it's much more prevalent in cases. So, but comparing 81% and 7% doesn't give you the all the information. We also did viral load. So basically, we found that the cases of those that are AAV2 positive had much, much higher viral loads, both in the blood and also in the liver. We also looked at the ISARIC protocol allows us to take longitudinal samples. So in the patients where we have longitudinal samples, we're able to measure viral load over time. And we basically show that, high levels of AAV2 doesn't hang around in the blood. And high levels seem to coincide with also very high levels of liver inflammation. So maybe when the patients are presenting with the jaundice and the liver inflammation, that's also maybe when AAV2 has very high viral levels. We then also looked at serology because that gives us, look at antibody response. So in the patients where we had residual blood left over, we did IgM and IgG, and we found that 2/3 of the patients, I think we had enough blood for 23 out of the 32 patients, and 2/3 of them were IgM positive. And you would think all of those should be IgM positive, but when we looked at those that were IgM negative, we found that 4 of them had their blood taken within 3 days of illness onset. So it's really important to have the clinical information, not just a sample, that the detailed history, and I can kind of expand on that a little bit later. And then 2 of the cases that were also IgM negative, had presented 77 days earlier, so 11 weeks sooner. That maybe by that time you'd lost your kind of acute IgM, and quite a lot of them, 91% of them were IgG positive. So at this point, you now have higher proportion of cases that have AAV2. Those that are positive in cases have much higher viral load levels. They're also demonstrating serological evidence of acute infection. But then we also looked within the liver as well, because that is clearly where the pathology is. And Vanessa can tell you all about what we did with the liver samples that we had.
This is literally where I was going to go to next. So, Vanessa, so you were doing some histopathology and some immunohistochemistry. And from what I can see in the paper, you've got some lovely images of the liver in there. And so my question kind of, I think it's the first one that anyone would think of when they're looking at this study is, how does the histopathology differ in these cases, or does it even differ from these children who are experiencing this AAV2 associated hepatitis versus someone who perhaps has a hepatitis C virus or some other form of liver disease? Is there any differences, or?
And this is, I think, the most important, and for me it was also the most interesting question. Because the first sample we received for histopathology was one of the patients who had the explant. So I had the sample from the explant, and the first look at the section showed that there are specific cells next to severe inflammation and bile duct proliferation. The hepatocytes showed a very ballooning phenotype. The cells were enlarged and they had a foamy cytoplasm. And this was the first thing which was very, very obvious. And then the question was, when we see this in this patient where the liver was transplanted, is this a phenotype which is true for all of the patients who are affected? Or is this something which is only specific for this patient? And it's very important to prove this, because if we have a viral infection, we always want to see a specific phenotype which comes with it as a proof of principle, basically. So we had this, and the question in the room was, after this first patient, is this true for all? And then we had more samples, and the other samples were these were not explant livers. These were all biopsies, so the sample was smaller, and the degree of inflammation and changes were not as severe. So, but the cells we were after were also present in the other affected ones. So it has a very specific phenotypes next to the inflammation, and depending on the degree of severity, we have inflammation. So the cells with the ballooning phenotype were always there in affected patients, and the inflammation, the degree of inflammation was varying, basically. And that was interesting, and which we wanted to prove with more samples during this study, basically.
And so just to check my own understanding, you're saying that the level of damage, if we want to call it that, seen within the liver corresponded with the was it the viral titer, or what was it correlating to, or
with this, but with a very, very severe, it needed a transplant.
Right. Yeah.
So some of the patients who had biopsies, they had mild inflammation. They were also severely ill because the liver cells showed morphology, which is an impairment of liver function. However, the damage was not as severe as in the explant livers. So this makes the difference. So we had in the explant liver, the patient has been treated for a longer time. We can see this in the morphology of the liver. There are cells which are only there if the liver is ill for longer. And this was very obvious in this particular case. And then the next question, which was really, really interesting for us, because as you have heard before, metagenomics PCR could show AAV2 nucleic acid in the blood and also in the liver. What was important for us to show where in the liver is the virus, what is which cell is affected. And the hypothesis before doing the experiment was, okay, the hypothesis is the hepatocytes show the ballooning phenotype. So this is something what we can see basically, and which is evident and which you can show everyone. Are these actually also the cells which are positive for the virus? Because for the blood PCR and the liver PCR, we cannot locate where in the cell is the virus. Is it actually the hepatocyte, or is it any other cell type? For example, the bile ducts, is it the inflammatory cells? So we ordered probes to detect this particular sequence of the AAV2 in order to find out where exactly is the virus located. And this was a very exciting experiment. And I can clearly remember that my boss sent me a Teams message every 5 minutes. Vanessa, do you have the results? Do you have the results? And the first experiment we did, and as you know, with new diseases and new experiments, you never know, even if you include all the controls, is this working? So the positive control shows basically a ubiquitous expressed gene. This worked. I checked this first, so I know the experiment did work, but is the virus actually there, and is the probe able to detect the genes? Yes, and it was. It was the most beautiful detection I think I've ever done, because the signal was so clear, and it was in 2 different cell types. So it was not only the hepatocytes with the ballooning phenotype, it was also in some of the endothelial cells of endothelial cells of arteria in the liver. So we had 2 cell types which are positive for virus signal, which was a very good result. And then we added more and more patients to the study, because it's important. If it's true for one, does not necessarily mean it's true also for the whole cohort. So all the patients we added showed that AAV2 RNA could be detected in the hepatocytes and the endothelial cells.
Amazing.
Yeah.
And I must admit, the images are very nice images. Not from someone who is.
It could be wallpaper, yes.
I was very like, oh, these are some nice images. So you see this ballooning phenotype within these hepatocytes. Is there any thought as to sort of the molecular cause of that on a molecular level, or is it just we know that the virus is there and this is the sort of phenotype that this causes, or is there any, or is that future studies is to look at what's happening?
So I think how exactly the virus interferes with the cellular mechanisms in order to impair the cellular function needs to be studied for sure. However, we need to keep in mind that the viral infection interferes with all the cellular functions, and if there is an impairment, the cell swells and the cell can't process properly, basically, and the bile will not be produced in the proper way. So this is in the first place a sign of a liver failure. However, the molecular details, I think, need to be studied more in detail. This is something we look into deeper. What we could do in parallel is to study the immune cells, which immune cells are associated with the disease, and this is what we did. And there are the other pictures in which you have seen in the paper that we studied T cells, B cells, MHC class 2 positive cells. So that's important to characterize next to the hepatic dysfunction on a molecular level, more also which immune cells are associated, to get an idea what kind of immune response or immune reaction do we have here. And this is what we also quantified to see what kind of cells are coming if we have an infection with AAV2. This was another part of the study, basically.
Amazing. And I think as well, speaking about immunology for just a little bit, there was something about the, and I'm going to get this wrong because I don't have it written down. It's HLA-DR somewhere.
DRB1
DRB1 0401. And this allele is to do with, and again, correct me if I'm wrong here, I believe it's to do with antigen presenting to, get your immune system going, basically, whenever it meets a new pathogen. You'll have presenter cells, which will engulf that, present it on the MHC, and then it'll be presented to other parts of the immune system. And you notice that this allele was present in, I think it was about 89% of the cases that presented with this. And so I've done a little bit of research on it, and from what I can tell is there's some association between that allele and potential autoimmune disorders. I noticed you also looked at different markers for autoimmune disorders, and it wasn't quite so clear what the connection there was. Has there been any further thoughts on that, or any ideas as to where that connection might fit in at all?
Yeah, I mean, as I said at the beginning, like autoimmune hepatitis is probably one of the kind of key differential diagnosis in children presenting with severe hepatitis, liver inflammation. But the markers that we check, which are a lot of them, we do routinely clinically, none of them, the children didn't really present with a lot of these autoimmune markers. So we felt that, and to say that, we recruited patients all across Scotland, and each of the centres had a paediatrician or a paediatric gastroenterologist, so someone who specialises in liver disease in children. It's not me. I'm an adult infectious disease doctor. But we kind of discussed all the cases that we recruited with kind of paediatric gastroenterologists in Scotland, and they didn't feel that the presentation or the markers fitted with an autoimmune hepatitis. So there was a case series that was published in Scotland of children with autoimmune hepatitis. Most of them were positive for autoantibodies, but also they tended to be older as well. So the epidemiology didn't quite fit. So as part of the kind of wide battery of tests that we did for all the kids, we looked at kind of whether host genetic markers were important, particularly as, the numbers were relatively small, because adenovirus infection, it's fairly common in kids, as is, AAV2 we talked about. Actually, majority, I think about 80 to 90% of adults are serology positive for AAV2, and we think that seroconversion happens in younger age. But why certain children? And to date, we've had 44 cases in Scotland and just over 270 cases in the whole country. Is it that you need to have some kind of genetic susceptibility that makes you manifest severe hepatitis? So yes, they turned out that this allele was very overrepresented in the cases. The HLA-DRB0401, which I'm only going to say once, was present in 93% of the cases. So I think 25 out of 27 cases that we managed to do the HLA typing for. And a lot of these HLA typing is, there's geographical distribution. So this allele is more common in North America and Northern Europe. And actually that fits with the kind of distribution of cases that have been reported worldwide. What you want to know is it something that in all people? And so what we did was we compared it to blood transfusion, blood transfusion donors, and we actually find that the background prevalence of this allele is only around 15%. So it's likely that this allele has implications, and you mentioned that you did kind of MHC class 2 staining, and there was definitely increased staining in the diseased liver, and it would fit with kind of a CD4 immune mediated immune pathology, and perhaps it's co-infection. So first of all, we think that this is likely to be a viral co-infection. We know AAV2 can't replicate by itself. It needed another virus, which is, adenovirus. We find adenovirus in a high proportion, but not all. Why do we only find it in smaller number of people? And a clue maybe, and this is purely what we're postulating, is that, because we take very detailed history from these kids, we find that over half of them present with a kind of gastroenteritic type illness between 1 to 11 weeks before they present with a hepatitis. And that's really important. We know that adenovirus, particularly the F41, can cause severe gastroenteritis. And maybe they presented with adenovirus in the first place. Because we don't recruit them right at the beginning, we don't really know which one comes first, and whether the AAV2 is reactivation or primary infection. We think it's probably primary infection, given that a lot of them are IgM positive, and the kind of age range that we see in under 5s is usually the age that children tend to present. It may be that, with why is it happening now? I mean, that's a very important question. Or last year, it's because we had very low levels of adenovirus for a long time, like all other respiratory viruses they were, they had very low circulation levels because of all the mitigation measures we had in place to prevent COVID transmission. So the kind of figure where we did the epidemic curve, you can see quite clearly that adenovirus, there was a huge spike of infection at the beginning of 2022. And that kind of epidemic curve really mirrored the hepatitis cases that we saw. And that kind of also kind of adds weight to this kind of co-infection. But not only do you need the viral co-infection, you also need to have this allele in order. So you have to have all three factors, a kind of triple hit phenomenon, in order to manifest this severe hepatitis. So it's a really exciting finding.
Yeah, absolutely.
Yeah.
And I think just to go back to a point you just made there about the SARS-CoV-2 and the lockdowns that we had, because I do know there, and we just have to address the horse in the room, the elephant in the room, because there was some speculation on Twitter that this was caused by lockdown, which I think is obviously not the case. This is shown otherwise, but I think there is maybe, how do I say this? You've got these kids who have been in the house for 2 years, not interacting, not picking up the usual viruses that are meant to when they go to nursery and play with all the toys that everyone's licked and slobbered over and all the rest of it, and picking up these little things. And so suddenly these, lockdown is relaxed, and then they're all mingling again, and then you're getting this spike in, adenovirus. Do you think that that's possibly why we saw this big spike all at once of 5 children coming together, or is that...
Yeah, probably. I mean, we looked back a number of years and we hadn't seen that many. I mean, all these cases aren't controlled for the volume of testing that we did, but we did see a huge peak in adenovirus infection that was about 2 weeks before we started seeing hepatitis cases. So the timeline fits. Compared to SARS-CoV-2, where we know there's been cases since March 2020, and I know there's been a lot of people who felt that this hepatitis may be due to SARS-CoV-2, or maybe kind of SARS-CoV-2 caused your immune system to react abnormally to another pathogen, for example, or some kind of co-infection. But we looked very hard for SARS-CoV-2. So from the kind of routine clinical testing, only 3 of the children were PCR positive, and 1 of them was PCR positive after they were admitted to hospital. So we don't think it's related. We don't think it's kind of direct SARS-CoV-2 related liver injury. We looked very hard on the metagenomic sequencing and didn't find SARS-CoV-2 on the metagenomic sequencing. And the other thing is we then look at the antibody response to SARS-CoV-2. If we think that it played a part, then we should find very high antibody exposure. But we only found it in just over half of the cases that had antibodies to SARS-CoV-2, compared to the background prevalence of antibodies for SARS-CoV-2 was, I think, between 59 and 72%. So we didn't find higher antibody prevalence in our cases. So we think it's very unlikely, but as you say, it may be that just because of the kind of non-pharmacological interventions from the pandemic and children not mixing, and generally they would all have over time gradually got adenovirus infection or AAV2, and because relaxation of these measures all happened at the same time, and kids and people started mixing, that we got this huge spike of adenovirus infection. And a paper that was published quite recently from Emerging Infectious Disease, actually from an Irish wastewater surveillance, illustrated this really nicely, that they looked at wastewater from one treatment plant that I think covers 40% of the Irish population. And they actually saw, again, the spike of adenovirus and AAV2 around the beginning of 2022, which is completely separate from our study. And so, the fact that another setting saw this in a totally different way in wastewater, which kind of illustrates the power of wastewater surveillance as well.
Absolutely.
And also there's another paper that we think is coming out from Nature at the same time from the UK HSA team and UCL, led by Judy Brewer, has also found very similar findings to what we found. So we think, that all the evidence that we found relates very well with other studies as well.
So is there anything else that's interesting about this allele that we've looked at, this HLA allele, that's different in these children who have presented with hepatitis?
Because I think it's quite interesting that we have the same HLA, DRB140, is also most prevalent in multiple sclerosis patients, and we are still unsure if it's a viral disease. It's interesting to see that we have virus infections which are more prevalent or more severe in some patients with a certain genotype. I think that's some aspect we can keep in mind for future research as well, to keep this in consideration when we study viral infection and why some populations or some people in the population show a more severe outcome than others. Because multiple sclerosis is also quite prevalent in Northern Europe, in Northern America, in this high latitude, low sunshine-ish countries, which I think is a nice coincidence to keep in mind, although causality is not proven. But I think this is where we might make hypothesis to keep this in mind, basically.
Okay, perfect. Thank you. And one thing that really came across when I was reading this paper is it feels very collaborative between clinicians, between public health bodies, between people in academia, between researchers on the ground, pipetting that stuff up and down, bioinformaticians. How was this project for you as a collaborative thing?
I found it to be a very positive experience. I mean, first of all, as a kind of clinical epidemiologist, I did a master's in epidemiology, and to kind of be involved in an investigation where you find, we're not saying this is the cause. We say there's this very strong association with AAV2 and adenoviral co-infection with this genetic susceptibility. But to be involved in a very comprehensive investigation that comes up with, a good hypothesis is really cool. It's pretty amazing. And I was genuinely I was just really impressed with how well, public health investigation, and that even within themselves, there are people, I attended these incident management team meetings where there's people from Scottish Government, from public health, but also from the laboratory, people who are doing the epidemiology, administering the questionnaire, and, to all the clinicians around the country who were, bending over backwards to help recruit these cases, and the research staff who, consent the patients and families to take part in the study, allow us to do research tests to these, we have my biobank manager, Sarah McDonald's fantastic, and she's the one who receives all the samples and then passes it on to the genomics team to do the sequencing, to Bioinformatician like Richard and the team who did all the analysis so quickly, and Vanessa and team looking at all the kind of immunohistochemistry and histopathology. It's been a really excellent collaborative effort, and I think driven by a really important research question. When you have this, in a way, it was a small, relatively small numbers, but it's a very severe presentation in that, about 5% of children requiring liver transplants and children who are really sick requiring hospitalization, which is huge. But for me, I think really great example of of collaborative research to address a really important public health question. I don't know if you guys have anything to add.
Yes, for me it was exciting because this kind of discovery research is what's all about in our work, I think. And this is a great fun, and I really enjoyed it to be part of this and being invited to bring my expertise, which is all about that we contribute with our work to do something better, actually. Okay. And what I really enjoyed as well, we had the chance to implement all the materials and techniques we have established over years before this, and could plug it in relatively quickly to come to a solution, to come as close to the solution as we could go, because we had what you mentioned previously, all the layers of adding layers of evidence. So we are as close to the proof of cause postulate as we could go. And this is what I really like, that we did everything we could do to come as close to the causality, although the causality is still to be proven. But this is actually our discovery research, what makes it amazing, that you work for years and then a study like this comes and you can instantly apply everything you have done previously to this, and relatively quickly it comes out and can give information to the public, to the patients, to inform people to be aware of what's going on here.
And for yourself, Richard, how did you find it?
I mean, there's two extra things probably to say. One is, I can't actually remember having a meeting in person everything's done by Zoom or Teams, which is pretty amazing when you think back. Like 3 years ago, if you said that, we're never going to meet in person and talk about it, you'd be crazy. But it was all Zoom, and there's lots of other kind of groups involved as well. So going back to that first set of 5 samples, we were asked to sort of share the data like immediately straight off the sequencer, and we shared that with public health bodies. So they were sort of essentially analysing at the same time as us. Then we had a meeting the next morning where we kind of say, well, what did you find? What did you find? And we found the same thing, which is like really reassuring that you're finding the same thing, which means there's nothing wrong with your pipeline, your analysis, all that kind of stuff. And then there's people in Edinburgh and HLAs, and then Judy's group as well. They ended up sequencing sometimes the same sample as well. It got sent, I think, down there, and they were finding the same things. So it's quite reassuring when everyone kind of converges on the same result. I think the Zoom thing's quite. I still find it quite surprising. it just worked. There was times when half of us were in the building, so you can see that the other person's at their desk at the floor below, but you're still talking about Zoom.
Zoom really has been the hero of the past 3 years.
But no, because again, that was one thing that really came across strongly in this, is how it was like, okay, immediately we have a problem, let's get everyone involved, and everyone pitched in and basically did their bit. And as you say, built these layers upon layers upon layers of evidence to come to what I think is personally a really strong conclusion of this very strong association between the AAV2 and this hepatitis that was presenting in children. So again, just want to congratulate you on the paper and the project, because again, I think this was fantastic to read. And with that, I just want to say thank you again for coming to be part of Research Goes Viral today. If you'd just like to go around and then give your name one last time, and then your
Can I just say thank you as well? I just want to thank Public Health Scotland and the MRC for funding this work. And also the funding from the MRC was done with ISARIC, which is led by University of Edinburgh and University of Liverpool. So Callum Semple and Kenny Bailey, who kind of co-lead this, and this infrastructure is really critically important to allow us to do all this work, and the work is not done. I think this is just the beginning for us to really understand, and we would like to do more work to really understand the kind of immune response underlying AAV2 infection. So we're looking at kind of pursuing more funding opportunities with that.
Cool. And just as one last thing, if you'd just like to go around and then your name and where people can find you, where you'd like to be found.
Where I'm likely to... Oh, right. Okay.
Like Twitter, mostly.
I'm Tony Ho. My Twitter handle is @Dr Tony Ho. My email address is also available on the CVR website as well.
Yeah, my name is Vanessa, and you can email me, obviously, with a Glasgow address, or on LinkedIn. It's easy to find. My first and second name is the same, so you will find me.
My name is Richard Orton. My Twitter handle is Richard J. Orton.
So once again, thank you all for joining us today, and thank you all for listening, and we will catch you on next week's episode.